For generations, one question has followed the Beta Israel of Ethiopia like a shadow. Are Ethiopian Jews descended mainly from ancient Jewish migrants who traveled deep into Africa? Or did Jewish identity develop among local Ethiopian populations and only later become connected to the wider Jewish world? The mystery became even more fascinating when scientists compared Ethiopian Jewish DNA with genomes from Ashkenazi, Sephardi, Mizrahi, Yemenite, and other Jewish populations, and discovered something that immediately separated Beta Israel from almost every major Jewish population they studied. Most Jewish communities formed a remarkably close genetic cluster with one another, and showed substantial ancestry connected to the ancient Near East and Levant. Ethiopian Jews did not. In genome-wide analysis, Beta Israel clustered primarily with neighboring Ethiopian populations, rather than with the major Jewish genetic cluster. That result did not mean Ethiopian Jews had no Jewish ancestry. It meant something much more complicated. Their biological ancestry was overwhelmingly shaped by the populations living in Ethiopia, while their Jewish identity developed and survived through a different historical process. Earlier studies of Y-chromosomes and mitochondrial DNA had already produced conflicting clues, with some evidence suggesting connections between Ethiopian Jews and other Jewish populations, while other markers placed them much closer to surrounding Ethiopians. The newest evidence made one thing clear. The genetic history of Beta Israel cannot be explained by a simple story of a Middle Eastern population moving into Ethiopia and remaining genetically isolated for centuries. Instead, the DNA points toward a deeply Ethiopian population whose Jewish identity became established through a complex history that may have involved conversion, male-mediated migration, and limited gene flow from other Jewish communities. And that single genetic result changed the entire way scientists approach the origin of Ethiopian Jews. To understand why this result was so surprising, you first have to understand what scientists expected when they began comparing Jewish populations around the world. For decades, genetic studies had found that many Jewish communities were more closely related to one another than to the non-Jewish populations living around them. Ashkenazi Jews, Sephardi Jews, Mizrahi Jews, Yemenite Jews, and several other communities often showed overlapping genetic patterns connecting them to the Middle East. This broad structure was consistent with historical evidence describing Jewish communities dispersing from ancient Near Eastern populations into Europe, North Africa, and Asia. Scientists therefore had a reasonable expectation that Ethiopian Jews might also sit somewhere inside that wider Jewish genetic cluster, perhaps showing a mixture of East African and Middle Eastern ancestry. Instead, the genome-wide results showed something substantially different. In the 2010 Nature study examining 14 Jewish diaspora populations, alongside 69 non-Jewish Old World populations, most Jewish populations formed a remarkably tight genetic cluster associated with the Middle East. Beta Israel was the major exception. Ethiopian Jews clustered closely with neighboring Ethiopian populations, rather than with the main Jewish genetic group. Indian Jews also showed strong affinity with surrounding Western Indian populations, although Bene Israel displayed a paternal connection to Levantine Jewish populations. The Ethiopian result was therefore not a minor statistical detail. It was one of the most visible differences among the Jewish populations examined. The researchers concluded that Ethiopian Jews had a distinctive genetic history that differed considerably from the pattern seen in most Jewish diaspora populations. But here is where the story becomes more interesting, because Beta Israel clustered with Ethiopians does not mean Beta Israel had no connection to the Jewish world. Genetics does not work like a membership card. A population can have predominantly local ancestry while preserving a cultural identity that arrived through migration, conversion, religious teaching, or a combination of all three. The genome-wide study was actually showing something much more specific. The biological ancestry of Beta Israel was substantially more Ethiopian than the ancestry of most other Jewish populations, was local to their surrounding communities. Ashkenazi Jews, for example, carry substantial European ancestry, but still retain a recognizable genetic relationship with Middle Eastern populations. Beta Israel, by contrast, looked overwhelmingly like an Ethiopian population in the genome-wide comparison. That difference became the center of the debate. If the community descended from a large Middle Eastern migration, why was the Middle Eastern genetic signal so weak? One possibility was that the founding group had been relatively small and later experienced extensive admixture with local Ethiopians. Another was that the community developed primarily through conversion of local Ethiopian populations, followed by strong endogamy that preserved the resulting Jewish identity. A third possibility involved smaller migrations of Jewish men into Ethiopia, followed by marriage with local women. The available evidence could not select one simple explanation with absolute certainty. Instead, it pointed toward a mixed historical process. This is why scientists have been careful not to describe Beta Israel ancestry as completely solved. The DNA has narrowed the possibilities dramatically, but it has not produced a single documented founding event. Long before whole genome sequencing became possible, researchers were already investigating Ethiopian Jewish origins using mitochondrial DNA and Y-chromosomes. These studies produced an intriguing contrast. Mitochondrial DNA follows maternal ancestry, while the Y-chromosome follows paternal ancestry. If a population was created by male migrants marrying local women, those two systems could look dramatically different. The mitochondrial DNA would be expected to resemble local Ethiopian populations, while Y-chromosome lineages might preserve stronger signals from the migrants. Some studies of Ethiopian Jewish communities did find patterns compatible with African maternal ancestry, alongside a limited Middle Eastern paternal connection. An early study of Jewish populations using Y-chromosome markers included Ethiopian Jews among several Jewish communities, and found evidence that Jewish and non-Jewish Middle Eastern populations shared an ancestral pool, but the Ethiopian community remained distinctive. Another study examining mitochondrial DNA across Jewish populations found African maternal types among Ethiopian Jews and noted that Ethiopian Jews were genetically distinguishable from other Jewish communities. These results gradually pushed researchers toward a model in which local Ethiopian ancestry played a very large role in forming the Beta Israel population. The important lesson is that one genetic marker can never tell the entire story. Maternal DNA might reveal a local origin, while paternal DNA might preserve a distant migration. Autosomal DNA then combines thousands of ancestral lines and can reveal how much of each population contributed overall. That is why the genome-wide studies were so important. They provided a much larger biological picture than earlier single lineage studies. The maternal evidence is especially striking. A study examining mitochondrial DNA from Jewish populations found that the Beta Israel carried a mixture of African maternal lineages that were common in Ethiopia, alongside some lineages associated with Western Eurasia. In the broader Jewish diaspora, many communities displayed maternal founder patterns associated with their own histories of migration and isolation. Ethiopian Jews were different. Their mitochondrial diversity looked much more like the surrounding Ethiopian population. Another study, focusing specifically on mitochondrial DNA in Ethiopian and Yemenite Jews, concluded that the Ethiopian Jewish population appeared to be a subset of the larger Ethiopian population, and suggested that its ancestry may have involved conversion of local women. This does not prove that every maternal ancestor of every Ethiopian Jew was a convert. It indicates that the population-level pattern was much more compatible with local Ethiopian maternal ancestry than with a large-scale migration of Jewish women from the Middle East. That difference is profound because it changes the picture of how a Jewish community can form. Judaism does not require every member of a community to share the same biological ancestry. A population can adopt a religious identity and then maintain it through generations of social institutions, marriage practices, ritual, and community boundaries. The Beta Israel case may represent exactly such a process. Their ancestors could have been predominantly Ethiopian genetically, while becoming Jewish culturally and religiously through historical processes that took place many centuries ago. The DNA, therefore, does not erase their Jewish history. It shows that religious identity and biological ancestry follow different paths. The paternal side introduces another possibility. Older Y-chromosome studies found that Ethiopian Jews shared some paternal lineages with other Jewish communities, creating evidence of at least some connection to Jewish populations outside Ethiopia. One early study proposed that Beta Israel may have descended largely from ancient inhabitants of Ethiopia who converted to Judaism, partly because several Y-chromosome haplotypes common among other Jewish populations were absent in the Ethiopian sample. Another later study found that Ethiopian Jews appeared genetically distinctive but showed relationships consistent with some interaction between Jewish populations and populations in the Horn of Africa. Taken together, these studies suggest that male-mediated migration cannot be ruled out. A small number of Jewish men entering Ethiopia could have contributed Y-chromosome ancestry while being absorbed into a much larger Ethiopian population. Their descendants could then preserve Jewish identity through community endogamy. Over many generations, the Middle Eastern genetic contribution could become small in the autosomal genome while remaining detectable in certain paternal lines. This is an important model because it would explain why the genome-wide signal looks predominantly Ethiopian while some paternal evidence points outside the region. But it remains a hypothesis rather than a proven historical sequence. Genetic markers cannot identify the ship that carried an ancestor, the village where he lived, or whether the original migration happened in one event. They provide probabilities about ancestry. Historical records and archaeology are needed to test the timing. Now consider Ethiopia itself, because the country's genetic history makes the Beta Israel mystery even more complicated. Ethiopia sits at a major crossroads between sub-Saharan Africa, the Red Sea, the Arabian Peninsula, and the broader Near East. Its populations have experienced gene flow in multiple directions for thousands of years. Studies of Ethiopian groups such as Amhara, Oromo, Tigrayans, and others have found varying amounts of ancestry associated with both African and Western Eurasian populations. The Horn of Africa has never been genetically isolated from the Arabian Peninsula. People crossed the Red Sea. Trade connected coastal regions with Yemen and other parts of the Middle East. Religious traditions also moved through these networks. This means that some Middle Eastern-related ancestry in Ethiopian Jews would not automatically prove a specifically Jewish migration. Researchers first have to distinguish Jewish-related ancestry from the broader Western Eurasian ancestry already present in Ethiopian populations. That is one reason the genome-wide comparison with many non-Jewish Ethiopian groups was so important. If Beta Israel had simply looked like other Ethiopians genetically, their Jewish identity could potentially have developed locally. If they had shown a much stronger Middle Eastern signal than surrounding Ethiopians, a major migration would have become more plausible. The actual result fell closer to the first scenario. Beta Israel clustered with nearby Ethiopian populations. That does not settle every historical question, but it strongly suggests that the community's biological foundation was local. The geography of northern Ethiopia and the Red Sea also provides an important historical backdrop. Jewish communities existed in and around the Middle East and Arabian Peninsula for centuries, and trade routes connected these regions to the Horn of Africa. Yemen, in particular, was an important point of contact. Genetic studies have found relationships between Ethiopian and Yemenite Jewish populations, although the interpretation is complex and may reflect indirect gene flow rather than a direct large-scale migration. Earlier research on an X-chromosome segment found that Ethiopian Jews appeared genetically admixed and identified evidence of a relationship with Yemenite Jews. The authors suggested that the connection may have resulted from indirect gene flow. This is an important distinction. A genetic similarity between two populations does not necessarily mean one directly descended from the other. They may have shared an intermediate population, exchanged genes through multiple migrations, or inherited related ancestry from an older source. The Red Sea was a network rather than a single road. People moved through ports, settlements, and trading communities. A Jewish ancestor entering Ethiopia could have come through Yemen, Egypt, Sudan, or another route, and their descendants could then have mixed into the larger Ethiopian population. Genetic data can identify the relationship, but archaeology and history are needed to reconstruct the route. The Genomewide 2010 Nature study provides the clearest large-scale comparison. Researchers examined genetic data from 14 Jewish diaspora populations and 69 non-Jewish Old World populations. Most Jewish groups formed a compact cluster in genetic space near Middle Eastern and Mediterranean populations. Ethiopian Jews were different. They grouped primarily with neighboring Ethiopian populations. Indian Jewish communities also tended to cluster with surrounding Indian populations, although Bene Israel showed evidence of paternal ancestry connected with the Levant. This result demonstrates that Jewish diaspora history produced different genetic outcomes depending on the community. Some groups retained substantial ancestral connections to the Middle East, despite centuries of life in Europe or North Africa. Others became more genetically similar to their host populations. The degree of genetic continuity depended on population size, marriage patterns, migration, conversion, and the amount of local ancestry introduced over generations. Beta Israel appears to have experienced particularly strong integration with its surrounding Ethiopian population. The result is not evidence that Ethiopian Jews are less Jewish genetically. It is evidence that their Jewish community was formed differently from Ashkenazi or Sephardi communities. Their history may have involved local conversion and limited migration rather than a large migrating population from the Levant. This distinction is exactly what genetics is capable of revealing, but why would a local Ethiopian population become Jewish in the first place? That is one of the biggest unanswered historical questions. Several theories have been proposed. Some traditions within Beta Israel connect the community to the ancient Israelites, sometimes through the tribe of Dan or other biblical narratives. Other historical interpretations suggest that Jewish practices may have spread into Ethiopia through contacts with Jewish communities in Yemen, Egypt, or the Red Sea. Another possibility is that local Ethiopian groups adopted forms of Judaism through religious exchange and gradually developed an independent Jewish tradition. Some scholars have proposed that Beta Israel emerged from a combination of local converts and migrants, followed by centuries of isolation from mainstream Jewish communities. Genetic data is consistent with the possibility of substantial local conversion, but genetics alone cannot tell us why people adopted Judaism or precisely when the process began. Religion spreads differently from genes. A group can adopt a religion without large-scale migration. Christianity, Islam, Buddhism, and Hindu traditions all demonstrate this repeatedly throughout history. Therefore, the absence of large, Middle Eastern genetic ancestry does not make the historical development of Ethiopian Judaism impossible. It may actually make a process of local conversion and cultural transmission more plausible. The genetic result forces us to think beyond the assumption that religious identity must arrive with a population replacement. This is where the phrase Beta Israel ancestry must be separated from the question of Beta Israel identity. A population can be genetically Ethiopian and Jewish at the same time. Jewish identity has never been based solely on a single biological marker. Jewish communities have historically incorporated converts and descendants of different populations, although the extent varied by period and location. The existence of Ethiopian Jews whose genomes cluster with neighboring Ethiopians therefore does not invalidate their Jewish history. It tells us that the biological and cultural histories are not identical. The community preserved Jewish traditions through religious practice, family structure, ritual, and collective memory. Genetic ancestry changed over time, but cultural continuity remained. This is one of the most important lessons of the entire study. DNA can tell us who people are biologically related to. It cannot determine which religion they belong to. A genome can reveal ancestry, but identity is a social and historical phenomenon. The Beta Israel are a perfect example of why those concepts should not be confused. Another important clue comes from the history of endogamy. Once a local Ethiopian population adopted Jewish identity and became a distinct community, marrying primarily within the group would have preserved the community's genetic and cultural characteristics. Endogamy can make a population genetically recognizable even when its original ancestry is mixed. It also allows rare genetic variants to become more common through founder effects. In Beta Israel, the genetic structure suggests that their community was relatively distinct from surrounding populations, even though their overall ancestry remained largely Ethiopian. This could happen through generations of limited intermarriage. The result is a fascinating combination. Genetically close to the local population at a broad level, yet socially distinctive enough to preserve a separate identity. This same pattern appears in many isolated populations around the world. Religion, geography, marriage rules, and political boundaries can all influence gene flow. In the Beta Israel case, social identity appears to have played an especially important role in maintaining community boundaries after their formation. Their DNA is therefore evidence of both integration and separation. Their ancestors became genetically Ethiopian, but their descendants remained socially distinct. The older mitochondrial studies make the story more detailed. One large study of Jewish mitochondrial diversity included 29 Beta Israel individuals, among a much larger dataset of Jewish and non-Jewish communities. The researchers found that the Ethiopian sample contained several African mitochondrial lineages, including branches that are common in sub-Saharan Africa, alongside some Western Eurasian lineages. This combination is very different from many Jewish communities outside Africa, whose maternal ancestry is largely Western Eurasian, despite varying degrees of local admixture. The Beta Israel maternal gene pool therefore provides another piece of evidence for deep local ancestry. But the presence of some Western Eurasian maternal lineages demonstrates that the population was not genetically isolated forever. Gene flow occurred. The exact timing and scale remain uncertain. This is why the population should not be described as either purely Ethiopian or purely Jewish Middle Eastern. The real genetic picture is mixed, with overwhelmingly strong local ancestry and smaller contributions from outside populations. The percentages and exact haplogroups matter less than the overall demographic pattern. A local Ethiopian population became part of the Jewish diaspora while retaining most of its regional genetic background. The comparison with Yemenite Jews provides another fascinating clue. Yemenite Jewish populations themselves have a distinctive genetic history shaped by their location between the Middle East and Africa. They carry substantial Middle Eastern ancestry, but also show evidence of African gene flow. When scientists find some genetic relationship between Ethiopian and Yemenite Jews, the connection may therefore work in both directions. It does not necessarily mean Ethiopian Jews descended from Yemenite Jews. Instead, the Red Sea may have allowed genetic exchange among populations on both sides for centuries. This is another reason the ancient world cannot be divided cleanly into Africa and the Middle East. The Red Sea was a corridor. Yemen and Ethiopia were linked through trade, migration, religion, and warfare. The same routes could carry people of different backgrounds. Jewish communities participated in these networks alongside Christian, Muslim, and local populations. The genetic result is therefore a shared regional history rather than a simple one-way migration. The story becomes even more interesting when we compare Beta Israel with Indian Jewish populations. In the same 2010 Genomewide study, Ethiopian Jews clustered with Ethiopian populations, while Bene Israel and Cochin Jews clustered with neighboring Western Indian populations. This means 3 Jewish diaspora populations developed very different genetic profiles depending on where they settled. Ashkenazi Jews retained strong genetic connections to the Middle East, while acquiring European ancestry. Cochin and Bene Israel became strongly Indian while preserving Jewish-related ancestry. Beta Israel became strongly Ethiopian. The pattern demonstrates a general principle. Diaspora populations adapt genetically to their local environments through admixture, while cultural traditions may persist through community boundaries. A diaspora is therefore not a frozen piece of the homeland. It is a living population created through interaction with its new environment. The Jewish diaspora produced many different genetic outcomes because each community experienced a different local history. This is why the Beta Israel result stands out. It is one of the clearest examples of a Jewish population whose genetic history became deeply integrated with the surrounding population. The most dramatic mistake would be to interpret the result as proof that the Beta Israel are simply descendants of Ethiopian converts with no connection to earlier Jewish populations. The evidence does not support such an absolute conclusion. Some paternal and other genetic studies have detected connections with Jewish populations outside Ethiopia, and historical contacts across the Red Sea make some degree of migration entirely plausible. What the Genomewide data shows is that whatever external ancestry entered the community was relatively small compared with the much larger Ethiopian genetic foundation. This allows a balanced interpretation. The community may have formed through local Ethiopian populations, adopting Judaism, supplemented by smaller migrations from Jewish communities elsewhere. Those migrants may have contributed disproportionately through male lines, while local women contributed much of the maternal ancestry. Later, endogamy preserved their religious identity and the resulting genetic profile. This model can account for the apparently contradictory evidence better than either extreme. It explains why Beta Israel resembles other Ethiopians genetically while retaining some connection to the wider Jewish genetic landscape. There is also an important archaeological limitation. Ethiopia has a long history, but the archaeological record associated specifically with early Jewish communities is difficult to interpret. A particular artifact may reflect trade rather than settlement. A religious practice may spread through cultural contact rather than migration. Written references to Jewish communities in Ethiopia become clearer during later historical periods, but identifying their exact origins remains difficult. This is where ancient DNA could eventually provide the missing link. If scientists discover well-preserved remains associated with early Beta Israel communities, they could compare those genomes with modern Beta Israel and surrounding Ethiopian populations. A direct ancient genome could reveal whether the genetic structure visible today was already present centuries ago or developed later. It could also show whether external ancestry entered repeatedly or primarily during one founding event. Unfortunately, ancient DNA preservation in Ethiopia can be challenging, especially in warm environments. This means researchers may have to rely on modern genomes, historical documents, archaeology, and computational reconstruction for some time. The mystery is therefore partly solved but not closed. Recent population genetic work in Ethiopia also shows why the region is uniquely important. A large Nature communication study examined the relationships between genetic similarity, language, geography, and religion across many Ethiopian populations. The researchers found that geographic distance and language both correlate with genetic structure, while cultural affiliations can sometimes reflect underlying population relationships. Ethiopia contains extraordinary linguistic and cultural diversity, including Semitic, Cushitic, Omotic, and Nilo-Saharan speaking communities. Genetic history follows some of those boundaries, but also crosses them. The Beta Israel are therefore part of a broader Ethiopian landscape where ancestry and identity frequently overlap without perfectly matching. Their Jewish identity represents one distinctive cultural branch within a genetically diverse region. Understanding Beta Israel origins requires understanding Ethiopia itself. A population cannot be interpreted against a blank background. It has to be compared with all the surrounding populations that could have contributed ancestry. The history of the Beta Israel also includes long periods of geographical and political isolation. Their communities developed mainly in northern and northwestern Ethiopia, where they maintained distinctive religious traditions and social organization. For centuries, they were not closely connected to the larger Jewish world. This isolation likely influenced their genetic structure. A population that remains relatively endogamous for many generations can preserve a distinctive genetic profile even when its original ancestry is mixed. It can also develop founder effects and unusual frequencies of certain variants. The combination of local ancestry and isolation makes Beta Israel genetically recognizable as a population distinct from neighboring Ethiopians, even though their broad ancestry remains closely related to those neighbors. This distinction is important. Genetic similarity does not mean identical population. Beta Israel can cluster close to Ethiopian populations while still showing genetic patterns associated with their own demographic history. The community formed its own genetic identity over time, just as it formed its own cultural identity. The story also shows why ancient Jewish history should never be reduced to a single biological narrative. Jewish populations around the world experienced different combinations of migration, conversion, admixture, and isolation. Some communities retained strong Middle Eastern ancestry. Others became genetically similar to neighboring populations. Some received later migrations from other Jewish communities. Others developed in near isolation. Ethiopian Jews represent the extreme end of this spectrum. Their genomes show that Jewish identity can persist even when most of the population's ancestry comes from the surrounding region. This is not an exception that invalidates Jewish population history. It is evidence of how diverse Jewish history actually was. The Jewish diaspora was never genetically uniform. It was a collection of local communities connected by religion, culture, and varying degrees of ancestry. So what exactly did scientists discover when they compared Ethiopian Jewish DNA with other Jewish populations? They discovered that Beta Israel stands apart. Most major Jewish populations studied form a relatively close genetic cluster connected to the Middle East. Ethiopian Jews instead cluster primarily with neighboring Ethiopian populations. Their maternal DNA contains predominantly African lineages, and their genome-wide ancestry is far more similar to surrounding Ethiopian groups than to Ashkenazi, Sephardi, Mizrahi, or Yemenite populations. At the same time, older Y-chromosomes and other genetic studies have detected some connections between Beta Israel and Jewish populations outside Africa. That combination suggests a complicated origin involving substantial local Ethiopian ancestry and a smaller external Jewish contribution. Studies have also suggested that local conversion may have played an important role, especially among women. The exact historical sequence remains unresolved, but one thing is clear. Beta Israel cannot be explained by a simple story of an intact Middle Eastern Jewish population migrating into Ethiopia and reproducing in isolation. The genetic evidence is inconsistent with that model. Instead, the most plausible interpretation is that Ethiopian Jewish identity developed within an Ethiopian population whose ancestors may have received limited genetic contributions from Jewish communities connected to the wider Near East and Red Sea world. Some Jewish migrants may have arrived, some local people may have converted. Some marriages may have connected Jewish newcomers with Ethiopian families. Over generations, the community became increasingly endogamous. Jewish traditions were preserved. The genetic profile became distinct from neighboring groups, but the majority of ancestry remained Ethiopian. This model explains why the Beta Israel looked genetically Ethiopian in broad genome-wide comparisons, while still retaining evidence of outside connections. It also explains why maternal and paternal studies do not always tell the same story. Different migration processes affected men and women differently. A few male migrants can leave a detectable paternal signature without transforming the entire genome. Local women can contribute most of the mitochondrial ancestry, while their descendants become fully integrated into the Jewish community. The deeper lesson is that the DNA did not reveal a simple answer. It revealed a process: migration, conversion, marriage, isolation, cultural transmission, and survival. The ancestors of Beta Israel were living in Africa. Some genetic connections reached beyond Africa. Jewish identity developed and endured, and the community became something that could not be reduced to either side alone. That is why the results stood out. The DNA did not say, these people are simply Middle Eastern Jews living in Ethiopia. It also did not say, these people have no connection to the wider Jewish world. It revealed something much more human. A community formed where cultures met. An identity survived after ancestry changed. And a population remained Jewish while becoming deeply Ethiopian genetically. That may be the real mystery of Beta Israel. Not where one group came from, but how a community can preserve an identity across centuries even when its genetic history becomes intertwined with the people around it. The genome can reveal the ancestry. It can reveal the migration. It can reveal the mixture. But it cannot erase the history people created afterward. And after centuries of isolation, change, and survival, the DNA of Ethiopian Jews is telling scientists something remarkably powerful. Their story was never a simple line from one ancient homeland to one modern community. It was a bridge between worlds. And the strongest clue was not the DNA that connected them to other Jewish populations. It was the DNA that revealed just how deeply they had become part of Ethiopia.